We try to take a lot of data analytic tools and systems and implement them for TB to improve patient care and really determine the role of diagnostics and the role that diagnostics have in patient care. Early on, before 2010, the type of diagnostics used for patient care was really a sputum. A person would produce sputum, put it on a microscope slide, and the sensitivity of that is really quite low. And a lot of people, specifically in South Africa, that are HIV co-infected with TB, have difficulty in producing sputum. So that test becomes even more insensitive.
The Expert MTB RIF, that was one of the game changers in the TB diagnostic testing world. It's basically a molecular technique, so it gives you a very rapid result within about 2 hours, compared to the diagnostic that was used before, that is TB culture, and that can take anywhere between 2 to 3 weeks before you actually get a result.
So it meant you got a test much quicker, more sensitive, and as a patient be able to be put onto treatment very quickly. And again, we were able to use our quality systems, our training platforms to be able to implement this. But the most important thing was really on the data side. Every single instrument in the field is connected to a single laboratory information system through the NHLS for public sector testing. So we took millions of results, 2.5 million every year, from the central data warehouse and analyzed it. We linked it down to a single geographic information system point where a patient provided their specimen, and were able to say where in South Africa testing was being performed, what the positivity rate was in South Africa.
So when I teach diagnostics, I always say, you want to think about the 3 A's of diagnostics, which is accessible, affordable, and accurate, right? And so that's what we were looking for TB. The work that was done in South Africa on TB, because TB burden is so high, has always sort of led the way in terms of evaluation of these new technologies, and then figuring out how to use them, and without incurring a lot of program costs, and how do you integrate it into other conditions that these TB patients may have.
So we want to look at something that's affordable, something that everybody can get access to. And then also time-wise, we want to cut around turnaround times. For instance, if I'm talking about TB, cutting down the turnaround time from 42 days of the midget to flag positive down to, let's say, 45 minutes with the gene expert. And also we want to look at something that is close to point of care for the patient. So something that is closer to them in the clinic they're being tested. Can we put this instrument there? Then as the patient comes in, they get their results, and then it's quicker. And then we cut on the infection spread rate.
Any developer, any company that has a new TB test comes to us, and they basically ask us to help either develop the test or to evaluate the test. And we start off with a lab evaluation. Depending on those results, if a test actually works, it gives you the result that you want from the assay. We then move on to a clinical evaluation. So this involves setting up a clinical trial, enrolling participants on this trial, and then getting samples from these participants. And if the test is found to be easy to use, it gives you the desired results, then these tests are actually implemented in South Africa, but also in many other countries around the world. So the work that we do is actually quite important because we are able to make these diagnostics available for the people that need them the most.
obviously we didn't know at the time of the Newton grant, was really the impact that all our data analytics skills had on COVID testing. So at the initiation of COVID diagnostics need in South Africa, we rapidly took our data team that was very well developed now on the Newton project for TB tests and TB data. We said, well, surely we can use the same digitization and granularity of testing for COVID. And we actually developed technology that enabled the scientific community in South Africa to see and predict the COVID waves at least 2 weeks before they happened, using purely the molecular data tools that we had developed on the Newton Fund. That's pretty cool.
After the onset of COVID, we started seeing a huge decline in the actual number of TB tests that were coming in. The data analysis that we performed showed that it was a reduction of 50% in the number of tests that were performed shortly thereafter. So it had a huge impact.
We really got to get back into all the lessons that we've learned, specifically through the Newton project, evaluating technologies, implementing technologies, and accessing patients. We want to really make sure that we get back to that after COVID for TB.
As soon as COVID hit, everybody or the world knew that we needed diagnostic tests and we needed them fast. And I mean, in a matter of a year, there's been hundreds of different molecular tests, antigen tests, and serology tests that have been developed. And I just think that the same kind of initiative needs to be applied to TB. Because I mean, it's possible in a short period of time, if you can develop all of these diagnostics for COVID, then why not for TB? There's a lot more work that can be done. It's very exciting. There's a lot going on out there. There's a lot more research that's going on, and yeah, I'm excited to be a part of that, definitely.